As I mentioned on the last presentation, cancer is directly related to the cell cycle and this whole issue of control and mitosis and cell division. And because so many people are touched by cancer, almost all of us probably know of someone who has had cancer or maybe passed away of cancer, I wanted to take a few minutes today and talk about cancer and how it directly relates to some of these issues that we're talking about as we think about cell division. So in particular, cancer is what we would call an acquired genetic disease. And this is very unusual because most diseases are either acquired, meaning they're infectious, you might catch it, something like pneumonia for example, or they're genetic, it's something you might inherit like cystic fibrosis. But cancer is different because it's an acquired genetic disease, and so it is a genetic disease, so it's a problem with the DNA in your cells, but it's not a problem that you've necessarily inherited. Instead, it's a problem that you've acquired, that over time your cells have developed these mutations or developed these problems. And genetically, the major issue with a cell that is a cancerous cell is it's a cell that's going to ignore those cell cycle checkpoints. It's not going to follow the normal rules of density-dependent growth. It's not going to stop growing when it gets crowded. It's going to ignore all the checkpoints such that your body cannot shut down that cancerous cell. Now remember, each of your cells has multiple checkpoints. And so a problem with one checkpoint, if you had a genetic problem or mutation, would not cause cancer. It would take all of those checkpoints to not function in the same cell for that cell to become cancerous. So for a cell to go from a normal, healthy cell that's following those rules of density-dependent growth that your body can control to a cell becoming cancerous, it takes multiple mutations of the DNA in the same cell. And so multiple mistakes or mutations. Again, one would not cause cancer because your cells have backup systems, several checkpoints. So a cell would have to have mutations or mistakes in all of the checkpoints to become cancerous. And so these mutations could be problems with the checkpoints themselves, such that maybe the checkpoints always send the go signal even if the growth factor is not present. It could be mutations in growth factors, such that maybe a cell makes growth factor all the time even though it shouldn't be. And so if that was the case, the growth factor is always present, the cell would always skip that checkpoint. Another major area where we see mutations associated with cancer are in our repair genes of our DNA. We know that mistakes or mutations in DNA are pretty rare, but when they occur, we've got a whole metabolic pathway of enzymes that recognize and fix those mistakes. But if your DNA repair mechanisms break and they don't work themselves, then you're more likely to develop other mutations in your checkpoints or your growth factor genes. And as we think of cancer, we all are kind of aware that while anyone can get cancer at any age, the older you are, the higher the risk of cancer, the more likely you are to develop cancer. And the reason for that is pretty simple. If you're older, your cells have been dividing longer. And every time your cells go through the cell cycle and you copy your DNA and you undergo mitosis, there's a small chance for a mistake. And so the more times your cells have copied their DNA and divided, the older your cells are, the more likely that these multiple mistakes might all accumulate in the same cell. So let's look at that idea of multiple mutations in the same cell. And again, it takes several mutations, a minimum of 3, probably more like 5 or 6 for different types of cancers, and they all have to be mutations in that same cell. And so if we look at our figure, on the left there we see a normal healthy cell. Let's say this cell has 4 checkpoints, and these different chromosomes represent 4 genes that are going to be controlling these different checkpoints. And it's much more complex than that, but this is a good kind of way to introduce our topic here. Let's say this cell is dividing, and it divides 100 million times. And every time it divides, no mistakes. Everything works out perfect. But in one of its cell divisions, as it goes through that S stage, it undergoes a mutation and it makes a mistake. And so that gets us to 1 mutation. Now, that cell's going to ignore 1 checkpoint, but it's still a pretty healthy cell. Your body can still stop its growth at 3 other checkpoints. That's not a problem. And it's only a mutation in 1 cell. But what's going to happen every time that 1 cell undergoes mitosis? It's going to copy its DNA, which now includes that mistake. And so every time that cell divides, it's going to copy the mutation. So while the mutation only occurred in 1 cell, if I came back 10 years later, you might have 500 million cells all with that mutation that came through mitosis from that original cell. Now one of those 500 cells now with that mutation 1 might undergo a second mutation. So now we've got a cell where 2 checkpoints work and 2 don't. Again, not a health problem, but that cell's going to keep passing on those mutations. So now if I come back 25 years later, maybe one of those descendants of that cell with 2 mutations has a 3rd mutation. Now we've got a problem. The cell's still not out of control. Sometimes we call this a precancerous cell because now there's only 1 checkpoint, 1 way to stop it. And so a final mutation in one of those cells then would result in a malignant or a cancerous cell. And so with 4 mutations, we would have a cell that's growing out of control. And so now your body has no way to stop it. And so here's how multiple mutations, might accumulate in a cell, usually over a very long period of time, to result in a cancerous cell. I've got a couple of examples of this multiple mutation idea here, and this is looking at colon cancer. And in this particular case, they compared the tumor cells that were cancerous, obviously, to the normal healthy cells of this person's colon, and they found about 5 different mutations along the way. And if we look on the left of the picture, that's the lining or the epithelium of the colon. Notice there's lots of space. It's a nice even layer of tissue. If we look on the far left, we can see the cancerous cells, and notice how tightly packed those tiny little cells are. They're really crowded and jammed in there because those cancer cells don't follow density-dependent growth. And if genetically we compare healthy to cancer, we can see this whole series of these 5 different mutations along the way that resulted in that cell becoming cancerous. We see the same thing with this example of a brain cancer, and this type of cancer is called astrocytoma, and astrocytes are just special nerve cells that help support your brain cells and keep them alive. And again, if we look at the healthy cell on the left, the malignant cells on the right, what we see is that there's a difference of 4 mutations here. And so these were 4 mutations that resulted in this brain cancer. And in this case, the tumor on the right there, you can see it up near the top front of the brain. Certainly this was a picture from an autopsy. This was most certainly fatal. And while we are very good at treating some cancers, for instance, breast cancer survival rates are up around 97% now, so we're getting very good at some cancers like breast cancer. Other cancers like lung or brain cancer, we have fatality rates up over 90 or 95%, especially cancers that are really hard to detect early. You can't really feel a lump with brain cancer or with lung cancer, and so they're much harder to detect. And here we see that tumor again up near the top. You're probably familiar with some common terms associated with cancer. A tumor is just an abnormal mass or a clump of cells that grow at a really high density. Again, normally cells grow in nice even layers, and so that's why you can feel a lump or feel a tumor if it's up near the surface of your skin, because you feel that dense clump of cells that normally don't grow that way. Now, nobody wants a tumor, but if we have a tumor and we get a biopsy, we hope it comes back benign, and benign would be a non-cancerous tumor. So those cells in that benign tumor are not mutated. They're not ignoring the checkpoint. It's usually not life-threatening. If you get the tumor removed, it's not a problem. Usually it's just due to some sort of small physiological problem. Maybe your body's making too much of a hormone, and that's why you got some cells growing faster than they ought to. If a tumor comes back as malignant, that's a cancerous tumor. Those are cells that are ignoring the checkpoints. They have acquired that genetic disease of cancer. And the problem with malignant tumors in terms of health issues is they tend to keep growing and they keep spreading as well. And all cancer starts out as a single somatic cell. So one of the cells in your body that develops these multiple mutations, and then over time that one cell divides into 2, and 2 into 4, and 4 into 8 by mitosis, and gradually over time that tumor is going to get bigger and bigger. And so here's just a picture of a cancer cell, a somatic cell, and your book has it kind of shaded. It looks very ominous and evil in this picture. But one of the big problems with cancer cells and why it's hard to treat is basically it's your cell. Genetically, it has an awful lot in common with all of your other cells, except for maybe 4 or 5 mutations in these checkpoint genes. And so when we think about treatments, one of the problems is how do we target and kill those cancer cells without killing your own healthy cells. Here we see a mammogram. On the left, we see a healthy tissue, a healthy breast tissue. Again, nice even layers, and things are kind of spread out there when we look at those cells. And we can see a mammogram of a breast with a tumor. We can see this kind of pea-sized tumor here. Certainly, again, that would be something you could feel because of that dense clump. Normal cells don't grow that way. And again, it started out as a single somatic cell, and it's estimated that to get to the size of maybe a pea-sized tumor that would contain around 5 or 10 million cells, that might take anywhere from 10 or 15 years from that single somatic cell to keep growing and dividing enough to get to that size. And so if we look and think about cancer, it's not that cancer grows really fast, it's that it grows persistently. There's no way to stop the growth or shut it off. So cancer cells are these cells growing out of control, which certainly begs the question, why is it so dangerous? Why is it such a deadly disease? And there's really 2 main aspects of the characteristics of cancerous cells that make it so dangerous. The first is what's called metastasis. And metastasis is just a scientific term for the spreading of cancer. And we know that while cancer will build up into a tumor, eventually that tumor will spread and that cancer will end up in other parts of your body. The other aspect of cancer cells is a process called angiogenesis. And anytime cells undergo mitosis in anywhere in your body, it takes a lot of energy. It takes a lot of oxygen, sugar, nutrients, and so forth. And so normally when cells undergo mitosis, they release hormones that cause blood vessels to grow to those cells and to feed those cells. So for example, if you broke your leg, the cells around that bone where they're going to grow new bone cells and heal would cause blood vessels to grow there, and that's associated sometimes, for example, with extra swelling. Well, these cancer cells are your healthy cells, and so they're going to release these same hormones, and they're going to cause blood vessels to grow to start feeding the tumor, which of course means you're going to have less food, less oxygen, fewer nutrients to the rest of your healthy cells. And so it's kind of a combination of this spreading of cancer and angiogenesis that's going to make cancer so dangerous to the healthy cells in your body. And in this picture we can just see the idea of metastasis. Again, if we think of something like breast cancer, a tumor, a pea-sized tumor there, is pretty localized, and if we catch it at this stage, we're very successful at treating it. And again, that's why there's so many warnings about self-exams and getting your mammograms and so forth, is because if we catch it before metastasis, we can treat cancer pretty well. If we catch it after, it becomes much more difficult to treat. And here we can see from 1 to 2 to 3 that cancer will gradually leave the tumor stage and start to spread into neighboring tissues. Eventually, if any cells get into your lymph system or into your circulatory, your blood system, they're going to spread to other parts of your body, and we may see secondary tumors in your kidneys or your lymph nodes or your liver, somewhere else in your body. And again, once it becomes a whole body disease, it's much, much more difficult to treat. One common misconception about cancer is that cancer cells grow really fast, and actually just the opposite is true. And here's some numbers, and you certainly don't need to know any of these numbers, but just for example, here's some healthy cells and the time it takes for these cells to go through interphase and divide by mitosis. You can see your bone marrow cells that make your red and your white blood cells. It takes a little under a day for cells to go through interphase and divide into 2 new daughter cells. For epithelial cells lining your digestive tract, whether it's your large intestine or your rectum or even your small intestine, it's usually about a day and a half to 2 days to get a new daughter cell produced. If we compare that to some cells that are cancerous, many cancerous cells actually take longer to go through interphase and divide. Here, the stomach cancer, about 3 days. Leukemias, which are basically cancers of the bone marrow or the blood, we're looking at maybe 4 or 5 days for that cell to undergo interphase and divide by mitosis. And oftentimes, because these cancerous cells have genetic mutations, they have problems with their DNA, it's going to take them longer to get through the cell cycle and divide than healthy cells. So it's not really the growth rate of cancer that causes the problem, it's the unrelenting nature of the growth that's going to cause the problems. That stomach cancer cell is going to divide every 72 hours from now on and not stop, versus that bone marrow cell, it may divide in 18 hours, but if you've got enough blood cells, your body can turn it off for a few hours or a few days until it needs more cells. And so many cancer cells actually grow slower than healthy cells. Now, cancer is a disease of mutations, and mutations are rare, but they're going to occur no matter what we do. If we lock ourselves in a room somewhere, we can still develop cancer, even if we're not exposed to anything that's particularly dangerous. But we know there are certain things in the environment that certainly can increase our risk of developing cancer. And any chemical that may increase your chance of getting cancer is called a carcinogen. So anything in the environment, and because, again, cancer is a disease of mutations, anything that might cause problems with your DNA would be a carcinogen. And that's where things like smoking comes in, radiation, UV light, your diet. We know eating lots of high-fat foods might increase your risk of mutation in your colon, for example. And so lots of different carcinogens out there. By avoiding carcinogens, you can reduce your risk of developing cancer. Now, we said cancer is a genetic disease. It's an acquired genetic disease. And so unlike other genetic diseases, if we look at inheritance patterns, you cannot inherit cancer, because it's a problem with your somatic self. For example, if I have lung cancer, obviously that's going to impact my health, but I'm not going to pass a lung cancer on to my offspring. I'm going to pass on my genetic material from my sperm cells, and so I'm not going to have to worry about passing those mutations on to an offspring. And typically, if we see cancer of the testes or the ovaries, that's going to result in sterility. My gametes aren't going to be produced very well or at all, and so I'm not going to pass that on. So we don't inherit cancer, but certainly the genes that we inherit from our parents can put us at either higher or lower risk. Certain genes may make us more likely to get cancer. Other genes may make us less likely to get cancer. And cancer certainly is a disease of risk. And one of the confusing things about cancer is I could be in a high-risk group and never get cancer. You could be in a low-risk group and develop cancer. And so there's no guarantees out there, but we can measure risk of cancer based on the environment and certainly based on your genes. And so there's a couple of genes that we know about that may increase your risk of cancer. The first couple discovered were looking at breast cancer, and they're just called BRCA1 and 2 for breast cancer gene 1 and breast cancer gene 2. And the idea here is that let's say it takes 5 mutations in your cell to develop breast cancer. If you already have one of these mutations, at this BRCA1 gene, for example, that means for you it would take 4 more mutations to get breast cancer. For someone without that genetic problem, their cells would take 5. And so all else being equal, you're more likely to develop 4 mistakes than someone else with 5, so you're going to be at higher risk for breast cancer. Again, you may never get it, but you're going to be at higher risk. Now, I do want to point out that over 90% of breast cancer cases are new or spontaneous, meaning they have nothing to do with genes, they have nothing to do with family. And so these BRCA genes are associated with only about 10% of all breast cancer cases. Most breast cancer are these spontaneous or new events. One other gene that seems to play a huge role in cancer is a gene called the P53 gene. And they've screened people with lots of common cancers like lung and prostate and breast and colon cancer. And what they found is about half of people with cancer, their cancer cells contain a mutation or a genetic problem with this P53 gene. Well, it turns out this P53 gene is the major gene that regulates your DNA repair system. And normally, if you have a genetic mutation in a cell, your P53 gene will cause your cell to stop at a checkpoint and give your cell a chance to fix that genetic mutation. If it's fixed, the P53 gene allows your cell to move on and divide by mitosis. If the DNA mutation is too major, if the cell can't fix it, that P53 gene will cause that cell to undergo apoptosis and it'll kill that cell off. The idea being your body's much better off losing one cell out of 100 trillion than it is passing on these genetic mistakes. And so people that have a mutation in their P53 gene cannot fix these mistakes. These cells pass on their mistakes, so they're going to be much more likely to have more mutations in their cells and in turn much more likely to develop cancer. If we move on to cancer treatments, and traditional treatments, again, for some cancers are very effective, like breast cancer and even skin cancer we're getting better at treating at. And the first gut reaction is, well, let's get it out, all right? Let's have a surgery, let's cut out that tumor. And certainly that's oftentimes the first stage, but not always. And one of the problems is we want to minimize the risk of spreading. If we're in there cutting around in your body, removing that tumor, if one cell falls off that tumor and falls into a new layer of tissue, we might have artificially spread or artificially caused metastasis to occur. And so we have to be careful there to minimize any risk of spread of that tumor. Now, other treatments are going to be aimed at killing off the cells that are in your body. And again, one of our main problems with our treatments is your cancer cells are the same as all the rest of your cells except for a couple mutations. And so how can we go in and target your cancer cells without killing all of those normal healthy cells? And that's pretty tricky business, and we're good at it at times and we're not so good at it with other treatments, and that's why there's oftentimes a lot of side effects with these cancerous treatments. The way that we're going to try to target cancer cells is we're going to try to target and kill off cells that are in the middle of mitosis. When cells are dividing in mitosis, they're kind of weak because they're spending a lot of energy. Their DNA is wrapped up in chromosome form, so they can't build any new proteins. And so if we can target cells in mitosis, we can hit a bunch of those tumor cells. Of course, lots of other cells in your body are undergoing mitosis too, and so that's why there's so many side effects with some of these treatments, is they're going to kill off those other healthy cells as well. And so chemotherapy, which is basically poison, we want to give you just the right dose or amount of poison to kill off cells that are weak by mitosis but not kill off healthy cells. An example of that might be a chemical, for example, like Taxol. And Taxol is a chemical that's used to treat a lot of common cancers, ovarian and breast cancer in particular. And Taxol is a chemical that will bind to the spindle apparatus during mitosis and cause the spindle proteins to lock. So that if a cell starts mitosis and you take Taxol, that Taxol will bind to the spindle and that cell will never be able to finish mitosis because it can't move the DNA around anymore. And so eventually that cell is going to run out of energy and die off. Another common treatment is radiation. Radiation is more of a localized treatment. We're going to add high energy to your body in very specific places, usually aimed at your tumor site, and we're going to give you just the right amount of energy to kill off these weakened mitosis cells and to leave healthy cells alone. Now, of course, either of these treatments, chemo or radiation, are typically repeated. You don't just get one dose, but especially chemo, you're going to come back maybe every 3 or 4 weeks over maybe a 5 to 8 month period, continually to repeat it. And the repetition, again, relate directly back to our cell cycle. Because every time you get a dose of chemo, it's going to kill your cancerous cells that are in mitosis along with your healthy cells in mitosis, but all of your cells in your tumor aren't going to be in mitosis at the same time. Many of your cells might be in G1 or S or G2, somewhere there in interphase, and those cells are not going to be killed off by your chemotherapy. So the idea is, if let's say 10% of your cells are in your tumor in mitosis today, if you get your chemo, maybe we can kill those cells and we can shrink your tumor by 10%. Come back in 3 weeks, a different group of cells are in mitosis. Maybe we can shrink it again by another 10%. And by repeating that process over and over again, we can gradually shrink your tumor and make it small enough that it's no longer going to be at risk of spreading or causing any health issues. So to wrap things up today, we know cancerous cells are cells that grow without control. They ignore the checkpoint system, and that's due to mutations. So it's an acquired genetic disease, and that it takes multiple mutations, 3 or more in the same cell, to develop a cancerous cell. Then we went over our cancerous terms about being benign or malignant, and talking about tumors and so on, metastasis, the spreading of cancer, the angiogenesis, or the feeding of the cancerous cells. Okay? And we know that cancer is a disease of risk. Some of that risk might be associated with your environment and carcinogens. Some of that risk is going to be due to inherited genes, and that our traditional treatments are pretty effective at treating most cancers, but certainly not all. Other cancers, again, like brain or lung cancer, still have a pretty high fatality rate.